Thank you, Magali, for that gracious introduction. Magali didn't point out to you that the reason I walk her and her colleagues to the elevator and often downstairs is it's a psychiatric hospital, and I'm afraid they might put those people away. So you heard this morning about the progress that has been made on the clinical front in understanding and treating post-traumatic stress disorder. But we have very little biological understanding of the underpinnings of stress disorder, and we have, as you heard from John Crystal, until now, no treatment of post-traumatic stress disorder. So a variety of approaches are necessary. And as you will hear from me and Ken Kosick and others in the next few lectures, animal models are one approach to this. I'm going to speak to you about one that we've been working with, which turns out to be really quite interesting because it's TIA, T-cell intracellular antigen 1, that is a sex-specific protective factor, and we've used this in a mouse model of post-traumatic stress disorder. There are two interesting features about TIA. One is that it's a functional prion that exists as both a solitary protein and as an aggregate. And two, that the aggregated form is a sex-specific protective factor of PTSD in mouse models. So let me speak about the first point, functional prion. Prions, as you probably know, were discovered by Stan Prusner in about 1982, and he characterized them in the following way. He said that proteins, these pathogenic prions, can exist in two forms, as a solitary protein, as an aggregate. And the conversion from one to the other is spontaneous. It's due to a misfolding problem. The aggregated form kills cells. And as it kills the cell, it is released by that cell, taken up by other cells, kills those cells, released by those cells, et cetera, et cetera. So this is a protein-only infectious mechanism that Stan Prusner described, and it's a new biological principle of disease. By accident, my colleagues and I, in 2004, discovered an aplasia, and then we confirmed this in the mouse in 2015, Kosick C. and Luana Ferrantini, that there are functional prions that have superficially similar properties, but they differ in two important ways. They exist as a soluble protein, as an aggregate, but the conversion is not spontaneous. The conversion is controlled. It's controlled by a modulatory transmitter. Serotonin in aplasia, dopamine in the mammalian brain. The aggregated form is functional, has a particular function in the cell. It's absolutely required. And the CPB, the first one we found, is a regulator of local protein synthesis at the synapse for long-term memory. As you know, with long-term memory, you grow new synaptic connections. Those new synaptic connections require proteins to maintain the growth, and those set of proteins are controlled by local protein synthesis regulated by CPB. So after we found this, I said to my lab, one example is a biological curiosity. Two examples is a biological principle. Let's see whether we can find a second example. And sure enough, Joe Raymond came up with this interesting example of TIA. This is converted from a soluble form to an aggregate form by glucocorticoids, and the aggregated form is a glucocorticoid receptor splicing factor, and it's a sex-specific protective factor, as I will show you, for post-traumatic stress disorder that really only works in female mice. So let me tell you something about its properties as a prion to begin with. It was Paul Anderson who first realized that TIA is an interesting molecule. It had a very characteristic prion domain, a glutamine-rich domain, and he suggested it might be a prion, but never really explored it further. We had experience, obviously, in looking at this, and one way to establish that it's a prion is to pop it into yeast and do some of the important tests that one can do there. And we found that all of the three major requirements were met by TIA. So TIA is self-perpetuating, so it forms these aggregates, and these aggregates can interact with the soluble protein and cause the soluble protein to be capable of forming aggregates. Number two, it's SDS resistant, it's resistant to protease, and it's heritable. It goes from one generation to another. The second distinctive feature, besides being a functional prion, is the aggregated form is a sex-specific protective factor for PSD in mouse models. Let me put this in a bit of a context for you. Post-traumatic stress disorder is really a model of a disorder. Classic is a gene-environmental interaction. This is true for most psychiatric disorders, but it's particularly profound here. If something very frightening were to happen in this room, most people would be unperturbed by it tomorrow, or the next day, or a week later. But 8% of men and 20% of women would come down with post-traumatic stress disorder. So most people would not, but a specific percentage of men and a larger percentage of women would come down with it. Presumably, this is due to the fact that they have some genetic predisposition to post-traumatic stress disorder. And this is exactly what one would like to know. What are the genes that are involved that are responsible for this? Clearly, in each case, it's not going to be a single gene. It's going to be a whole bunch of them. But to identify even one of them would be a useful beginning. So what's interesting about this is what are the genes? And specifically, why is it that women are so much more susceptible? 20% compared to 8%. And since very few genes have been identified in people, we turned to mice and focused first on TIA, used that as a gene that we studied, and the environmental condition we used is fear conditioning. TIA is present at low levels endogenously in the hippocampus. Its level increases in response to stress, and in the hippocampus is SDS resistant. It forms these aggregates, and those aggregates are also produced by cortisol, which is released by stress. I'm going to show you a series of experiments we carried out in which we carried out contextual fear conditioning. So contextual fear conditioning involves putting an animal into a chamber that has two components to it. One has a particular odor, and the other one does not. You shock the animal in the part of the chamber that has an odor. The animal will then remember almost anything related to that. The odor by itself, without the context in which it was shocked, everything related to that. So let me show you sort of a typical experiment. When you train animals to contextual fear conditioning and test them afterwards, in an open field, elevated plus mace, or conditioned odor avoidance, you find there's no difference between the wild-type mouse and the mouse in which T has been knocked out. So let me just tell you what these tests are. The open field is the animal avoids the open field in general. It's frightened of being exposed. In elevated plus maze, animals like to be in the closed arms, and in conditioned odor avoidance, it avoids the chamber in which the odor was present in which you were shocked. But if instead of looking just immediately, you look several weeks later, you find that now the knockout mouse is much more affected than the animal, the wild-type mouse. So it spends even less time in the open field, it spends more time in the open arm, and it avoids even more the side of the chamber in which the odor was, in which it was exposed to stress. So clearly you see all of a sudden a sort of post-traumatic stress disorder appearing, and is particularly profound in the knockout mouse. What is interesting is, if you look at male mice, you don't see this. the wild type and the knockout respond exactly the same. So this seems to be a female-specific protective factor. You knock it out, and the animal responds more profoundly. So what is the physiological basis for this? Well, obviously, there are going to be a number of different mechanisms that contribute to it, but one that really struck us immediately was a change in hippocampal function. There is, in female mice, a protective factor. As you know, hippocampus is involved in memory, and painful memories are also mediated by it. But in female mice that are fear-conditioned, the memory mechanism is decreased. So they don't have to remember this powerfully. And you can simulate this with cortisol. It also decreases the LTP, and this can be blocked by specific inhibitor of the cortisol receptor. If you knock out TIA, that protective step is lost, both for just plain LTP and also for the cortisol effect. The interesting thing is, again, male don't show this effect at all. So they don't show it to begin with, and the knockout has no effect on them at all. So further evidence that this is a sex-specific protective factor. So in other cells of the body, TIA serves as a splicing factor for the glucocorticoid receptor, and therefore we were curious to see whether or not it also serves as a splicing factor for glucocorticoid receptors in the hippocampus. Simply to remind you, in the classical glucocorticoid receptor signaling pathway, the glucocorticoid receptor GR-alpha, which is the canonical receptor, is bound up by chaperones in the absence of a ligand. When cortisol enters the cell and binds to GR-alpha, it frees itself from these chaperones, and then it forms dimers. And these dimers are of two sorts. It can form a homodimer with GR-alpha. This is the active, effective form of the dimer. Or it can form a much less effective dimer when it forms a partnership with GR-beta, which is a dominant negative inhibitor of GR-alpha. So there are two kinds of dimers, GR-alpha, and GR-alpha beta, and they differ greatly in their efficacy. So we're curious to see how TIA1 regulates the splicing of GR-beta in the hippocampus. And we found that it does it in a sex-specific manner. So GR-alpha and GR-beta are homologous isoforms that differ only in their C-terminal region. They're generated by alternate splicing of this region, intron 8 of the glucocorticoid receptor. Males and females have both forms, but TIA regulates the splicing in a sex-specific fashion. So both males and females have both forms, but if you knock out TIA, you see that GR-beta increases in the females, both the messenger RNA and the protein. There's no change at all in GR-beta in the male. Since this is so important, we wanted to check this with an in vitro splicing assay. So we just took this segment of the message, we incubated this with in vitro transcribed gRNA with nuclear extracts from wild type or knockout of the hippocampus. And again, we saw exactly the same thing, an increase in GR-beta, just where I showed you in the hippocampus, so we see it in in vitro splicing assay. Now, to really show that GR-beta is really responsible for this phenotype, we actually looked at the physiology. What happens if you overexpress GR-beta? Can you produce what you see when you have normal functioning of GR-beta? So we overexpress GR-beta in the dominant negative inhibitor of GR-alpha in the ventral hippocampus. And here we simulated what you see we took a knockout and we could restore the protective function that we normally see with cortisol. So let me summarize what the function of the GR-alpha and the GR-beta is. Under normal circumstances, you have a very good balance between GR-alphas and GR-betas, and that gives you an optimal response to stress. If you knock TIA out, you get more GR-beta, and this is much less effective in MAP kinase signaling, activating CREB, and the downstream effects that are mediated by that. So we're interested in seeing whether we can begin to get some insight into the structure of this. One doesn't have an insight into any structure of either functional or pathological prions. So we're starting to do this, and we're beginning to get some clue that we can maybe get a handle on the structure. We've identified as a pentameric structure, and we're going to do cryo-EM on its future, but in the meantime, just with negative staining, Chan Ling in my lab has been able to show this very beautiful pentameric structure. And this suggested to Joe Raymond, who's really the driving force behind these experiments, maybe we can get drugs that cause either aggregation or disaggregation of this. Now imagine if we had a drug that caused aggregation. What would we expect? Enhanced protection of female mice. If we called disaggregation, we would have the opposite effect, like a knockout. So Joe Raymond developed an in vitro FET assay that allowed you to see whether or not TIA was aggregated or not, and then to see whether you can develop drugs that, for example, would increase aggregation. After a while, we're going to try to, in fact, we're beginning to look at drugs that also decrease aggregation. And the way this works is you take TIA and you divide it into 2 batches. One you fuse to CFP, which is the donor. The other you fuse to YFP, which is the acceptor. And only when 2 TIA molecules are touching each other will the fluorescent signal be activated, so you'll be able to detect it. So in these circumstances, you can see in the basal level, you don't see any signal. But with this compound that he searched out, he went through a lot of compounds, but one particular compound, dose A, shows you a nice increase in the FET signal, indicating this increases aggregation. So here we have a drug that increases aggregation, and when you look, you in fact see that compound A causes a rapid formation of a high molecular weight SDS-resistant compound, very much like you see with conditioned fear or with cortisol. So this clearly is independent evidence that aggregate formation is occurring. So now we can look and see whether or not this is protective. I earlier showed you what happens under normal circumstances when you have a knockout, looking in this particular part of the paradigm, that is the conditioned odor avoidance. But after giving this compound, you see that in fact the animal, which spent very little time in this chamber, now that it has this protective factor, feels much more comfortable and is spending significantly more time in the chamber. So clearly this is acting as a protective factor. It's no longer as frightened. It's willing to tolerate being there for a longer period of time. As I said, this is a classic example of this, and we really want to get sort of a better understanding of it, and we want to move into new directions. TIA is clearly one of the genes that contributes to the difference between females and males in mice, and we also, as I will tell you, beginning to see whether or not this might be present in some human populations as well. So let me indicate to you the new directions we want to go, because this is really just the beginning. There's an enormous amount of work that needs to be done with this model and also with other models, because no single model is going to be sufficient. Number one, so far we focused on the hippocampus, but as you heard, there are a number of different structures involved. The amygdala is importantly involved, the ventral hippocampus, which I talked about, the prefrontal cortex. So we want to look at each of these structures to see whether they also show some evidence of post-traumatic stress disorder, and to what degree TIA is important there, or what other genes might be important there. So we want to characterize the neural circuit in the mouse that is involved in post-traumatic stress disorder. We're not alone with this. Other people are looking at this as well, but this is very important, because I think one thing you can do as a minimum is work out for this kind of post-traumatic stress what the neural circuit is. In addition, we want to not only examine the neural circuit, but we want to see the interaction between TIA and each of these areas. Is each of these areas, the physiology, also affected by TIA? Also, we're looking for other genes, certain genes specifically active on the hippocampus and on other regions, or do they all act on the same targets? And then we want to see whether we can begin to see this in human populations. So we've started 3 kinds of collaborations. One is with Murray Stein, who has a cohort of pre- and post-active duty deployment military personnel, and we have don't have any results from that as yet. Then with Myrna Weissman, we're looking at a family-based anxiety depression cohort, and we have a long-term Swedish longitudinal epidemiological study that we're collaborating with. So from the first study, as I indicated, the data are pending. The preliminary results from the second study indicate a significant interaction between TS-SNPs and childhood adversity in development of anxiety and depression. And in the Swedish longitudinal study, we again have data suggesting statistically significant interaction between TS-SNPs and stress for life events. We have not yet looked at a population that is specific for PTSD, but we plan to do that as well. Now, I should tell you, it's highly unlikely that one gene is going to be involved in all forms of PTSD, and I think Magali has gotten some recent evidence from the Broad Institute has looked at this, looked at a population in which this was not involved. This is not terribly surprising. It's very likely that a number of different genes are involved, and in any population, one particular gene might not be critically involved. So we are hoping that we get some data on this and get an idea under what circumstances it's most likely to be involved. So let me end by simply telling him Joe Raymond was the driving force behind this. Jan Lee did the structural study, and Irwin Ashok is doing the future experiments, and Philip Miller's hacked out on some of these critical experiments. And these are my collaborators. So thank you very much. I appreciate your attention. I'd be delighted to attempt to answer. I won't say I answered them successfully. Any questions that you might have? Yes?
Thanks very much, Dr. Kandel. So the problem with the epidemiological information, which sort of very attractively led to the strategies that you used here, the most recent epidemiological studies suggest that when you hold constant the kind of events to which people are exposed, as much as that is at all possible, for example, in the military in Afghanistan and Iraq, the numbers between men and women in terms of the prevalence of PTSD are consistently coming out to be the same. So that exposures are comparable and prevalence of PTSD is comparable. The difficulty with the 20% down to 8% in men is that it really confounded the kinds of things to which men and women are differentially exposed. So I'm kind of curious, is it, in my mind, there shouldn't be sex differences.
There are, at least in this case.
But clearly this is. So what are your thoughts?
What you're saying is interesting. I was not aware of that. But this clearly shows that there are sex differences between them. So maybe under certain circumstances that we happen to be tapping in on, you pick them up. But that's a very good point.
It's an honor to hear you speak, Dr. Kandel. I've heard of your name so many times over the years. I do want to ask a question in terms of, it was raised earlier that the animal models had not generally predicted success in clinical trials, but they have a power to go after molecular targets that we don't have with other methods. So one of the solutions that was mentioned earlier was whether or not we should choose outcomes that are translational outcomes, meaning the animal model, or not the animal model, but the behavioral outcome that you measured is not going to be one that we can test in clinical trials. So I'm curious, just from a more broader perspective, are there specific maybe behavioral tests that you think would be more easily translatable into the clinical trials?
Yes, I've thought a lot about this because this is clearly an important issue. Why are a number of mouse models not successful in giving you drugs that are useful in people? And to me, this has always seemed quite clear. Mice are not good models for many psychiatric disorders. There is no satisfactory mouse model for depression. The things that are tested are stress-related. There obviously are no satisfactory mouse models for schizophrenia because, how do you know whether a mouse is hallucinating or is delusional? So there are aspects of the disease you can test. But there are some diseases or some conditions that you can test beautifully in the mouse. One thing I've become interested in, not a moment too soon, is age-related memory loss.
We found that.
Scott Small and I've been looking at this in parallel. He's been looking at age-related memory loss in people. I've been looking in the mouse. They're indistinguishable. They both involve the dentate gyrus. They both involve similar molecular molecules. Obviously, we've been able to define them much better. The same kinds of things that reverse them in the mouse will reverse it in people. So one can really make a lot of progress in age-related memory loss with mouse models. The other thing they share in common is fear. You can study fear in all animals. So I think post-traumatic stress disorder is something one can study. One may have to modify the paradigm. One may want to agree which of the paradigms that are most useful. But I would be surprised if this did not turn out to be a useful animal model. We just need some Cohen consensus on what are the best paradigms to use.
Senator Collins and Senator Warren work in conjunction in a bipartisan way on the Committee on Aging, and they've earmarked some studies in NIH on aging. I don't know if you've looked at that, but it might be helpful. It's about gender differences in the brain with regards to aging. I don't know if that would be helpful to what you're doing, but
I'm sorry, I didn't hear what you said.
Senator Collins of Maine and Senator Warren of Massachusetts work together on the Committee on Aging in the Senate, and they've earmarked funds at the NIH to study age-related differences in the brain. I just wanted to mention that earmark to you.
But this is based on Scott Small's work, who first picked up there is a change in the dentate gyrus in age-related memory loss, as everyone knows, in the entorhinal cortex is beginning of Alzheimer's disease. When age-related memory loss first began, let me just step back a little bit. When I was a medical student, we never spoke about age-related memory loss, and we never spoke about Alzheimer's disease. You know why that's so? People didn't live long enough to get this. We saw patients born in 1900. In 1900, the average life expectancy was 50 years. Now, the average life expectancy is 78 for men and 82 for women. So you're seeing Alzheimer's disease, which is, like 10, 15% in 80-year-olds and almost 50% in 90-year-olds. And when Alzheimer's disease became more prominent, people weren't sure whether or not this other entity, which was also there, was an early form of Alzheimer's disease or an independent entity. Now the evidence is pretty clear. There is an independent entity, age-related memory loss. It has a different site of origin. It responds to different therapeutic maneuvers, et cetera, et cetera, et cetera.
It's more comment. First of all, thank you very much for beautiful exposition. It's very interesting. I'm more trying to address the comment about animal model and mice models in general. We had a couple of comments earlier today. One is, of course, important comment that many mice models did not produce useful strategy. On the other hand, there was also a critical comment which mentioned that some of those trials did not think about this animal model, mice model, didn't use it in a thoughtful way. There was problems with the trials. So I think it's important, as the field matures a little bit, is to not to throw away the baby with the dirty water. So models are critical for us to understand. We're just starting understanding, and I think maybe tomorrow we have more chance to talk about, that the models are not models of the syndrome or disorder. The models, we can model circuits. And as Dr. Kandel just mentioned, particular function associated with circuits such as conditioned fear abnormalities that he's studying. Clearly, what our role as translational scientists will be is to demonstrate in which part of PTSD, in what type of patients, in what type of symptomatology, these specific circuits and these specific functions are involved, and how can we treat it. It doesn't mean that in all of them it might be, maybe only in part of them. Maybe some of the PTSD will have a different circuitry abnormalities and some different entry into the same circuitry. So the mice models are or animal, mice, rats, monkey, all animal models allow us to actually interrogate those circuits and those functions ecologically meaningfully way, not just face value saying, oh, because humans don't freeze when they are scared, but mice do. We do understand that, and we measure fear in mice and human differently because we're different species. So when we understand and approach that carefully with taking model of circuit associated with function, which are ecologically translatable, I think, like the example that we were hearing right now, it will make a great progress, and I hope we'll continue doing it.
I think you're absolutely right, and you put it very well. I think I would just add two things. John Crystal, in his comments from the stage in the panel discussion, pointed out to a marvelous insight he got from a mouse model. And I think the second thing is, I think one of the things we can accomplish here is to define which particular tasks are particularly useful to examine in which particular, animal model. For example, maybe rat would be a better example for some. We can now do genetic experiments in rat quite well. We can get certain, even monkey models that one can begin to use. So we're not just limited to mouse. We use mouse because it's the most readily available, until recently, the only one we could really use genetics on. So I think to give up on animal models would be a serious mistake, but to use it more appropriately would be a very good mistake. And I think it would be a very good idea. And I think we are in a very good position to define exactly what are the best set of paradigms to use.